All right, this is Matt Dean with A Plus College Ready, and this is a review video for the AP Biology exam about statistics. Statistics are becoming a more and more important part of the AP Biology exam. You need to know how to do two important types of statistics. First of all, we have descriptive statistics. These are used to estimate certain parameters of our data set. Examples include things like the standard deviation, which tells us about the variability of our measurements. Also, the measurements of central tendency, like the mean, the median, and the mode. Also, we have the standard error of the sample mean, which helps us to determine how well our sample mean represents the real population mean, the broader group mean. In addition to descriptive statistics, we also have inferential statistics. And these are tools or statistical tests that use probability theory to essentially allow us to determine precise estimates of our true population parameters from our actual sample data. So we have a small sample, and this allows us to figure out how well that small sample fits the entire population. These are really important when we're doing data analysis, and these inferential statistics allow us to support and draw conclusions from our data about the overall true population. We mentioned standard deviation a minute ago. Standard deviation, you might see it represented with a sigma for the population. In terms of AP Biology, we're going to usually be doing statistics on a sample. So for sample standard deviations, you're usually going to see it notated as an S. Remember that the standard deviation tells you essentially how much each data point varies from the mean. A standard deviation near zero indicates that the data points are very close together, while a bigger standard deviation indicates a wider range of values, a more scattered distribution. Then we also have the standard error of the mean, which you might see notated as SEM. You might also see it notated as SEX bar. Essentially, the standard error is an estimate of how far the sample mean is likely to be from the population mean. So we're going to look at an example problem, and we're going to learn how to do some calculations here first with descriptive statistics. So it says you collected the following data in an experiment. During the experiment, you measured the height of plants grown at different fertilizer concentrations. So 20 grams per liter of soil, 40 grams per liter of soil, and 60 grams per liter of soil. And we measured the heights of 5 plants that were grown at each concentration. So we have 3 samples. So what we want to do for each group is we want to calculate the mean, we want to calculate the standard deviation, the standard error of the mean, and then we want to create a bar graph that illustrates the means to within a 95% confidence interval. And then we want to make some conclusions based on that information. So let's first do a little calculating. Let me find a blank slide that I can work on here. Let's work from here. So our 20 gram per liter sample, let's start with it. We're going to make a chart. We're going to make XI be our individual data points. So they were 15, 18, 22, 10, and 14. We know to find the mean, we simply need to add those together and divide by the number of data points, which is 5. So when we add those up and divide by 5, we get a mean of 15.8. The next thing we want to do is calculate the standard deviation. So to do that, we're going to create a chart. And our chart is going to look like so. The formula for standard deviation looks like this. It's the square root of the summation of each individual data point minus the mean squared over n minus 1. So that's the formula we want to work from. So to get to there, we're going to use this chart. We're going to go each individual data point minus the mean. So for this first area right here, it would be 15 minus 15.8. So we get negative 0.8. And then we're going to square that negative 0.8 over here to get 0.64. So now it'll be 18 minus 15.8. to get 2.2. And that squared is 4.84. Next one will be 22 minus 15.8 to get 6.2. 6.2 squared gives us 38.44. For the next one, 10 minus 15.8 to get negative 5.8. And negative 5.8 squared is 33.64. And then finally, 14 minus 15.8, negative 1.8. And negative 1.8 gives us, squared gives us 3.24. So to calculate the standard deviation, we're first of all going to find the summation of xi minus x bar squared. That tells us essentially just to add up this column. So the summation of this column, when we add them up, 80.8. So then we need to divide that 80.8 by n minus 1. N is our sample size. And in this case, our sample size is 5. So we're going to divide that by 4, n minus 1. And we're going to take the square root of the whole thing. So that's going to give us a standard deviation, once we do the calculation here, of 4.49. Finally, we want to calculate the standard error of the mean. And that is equal to the standard deviation divided by the square root of the sample size. So in this case, it's going to be 4.49, the standard deviation, divided by the square root of 5, 5 being our sample size. Calculate that out, and we get a standard error of the mean of 2.01. All right, so now we're going to complete the same calculations for the 40 gram per liter sample. So for that group, once we get this erased, our data points, our individual data points are 18, let's make a line there, 18, 24, 26, 22, and 16. So we add those up, and we're going to divide by 5 our sample size to get a mean. And when we do that, we end up with a mean of 21.2 for the 40 gram per liter sample. So now we fill in our chart to get the standard deviation. We're going to go 18 minus the mean, 18 minus 21.2 to get negative 3.2. We're going to square that negative 3.2 to get 10.24. Now we're going to go 24 minus 21.2 to get 2.8. We're going to square the 2.8 to get 7.84. Next, we'll go 26 minus 21.2 to get 4.8. We're going to square the 4.8 to get 23.04. Next, we'll go 22 minus 21.8 to get 0.8. We'll square that to get 0.64. And then we'll go 16 minus 21.2 to get negative 5.2. Square that to get 27.04. Remember, to calculate the standard deviation, we need to take the square root of the summation of xi minus x bar squared all over n minus 1. So now we need to sum up this column, because that's xi minus x bar squared. So when we sum up that column, that gives us 68.8. So to get our standard deviation, we're going to have 68.8 divided by n minus 1. So our sample size is 5, minus 1 would be 4. We'll take the square root of that whole quantity, and we end up with a standard deviation of 4.15. So if we compare the standard deviation of the 40 gram per liter sample to the 20 gram per liter sample, we see that the standard deviation is a little bit lower for the 40 gram per liter sample. What that tells us is that the samples were, their heights were a little closer together. They were a little more centered around the mean in the 40 gram per liter sample than they were in the 20 gram per liter sample. If we want to calculate, all right, so again, to calculate the standard error of the mean, we're going to go standard error of the mean, is equal to the standard deviation divided by the square root of the sample size. So for the 40 gram per liter sample, we're going to take our standard deviation of 4.15, and we're going to divide that by the square root of the sample size, which is 5. And that gives us a standard error of the mean of 1.85. All right, we need to complete these same calculations for the 60 gram per liter sample. So for that setup, our individual data points for the 60 gram per liter sample were 28, 26, 24, 18, and 29. So we need to go xi minus x bar, xi minus x bar quantity squared. First of all, we need to calculate the mean. So we add up 28, 26, 24, 18, and 29, and divide that by the sample size of 5 to end up with a mean of 25. Now let's fill in our table. So we're going to go 28 minus the mean of 25 to get 3, 3 squared to get 9, 26 minus 25 to get 1, 1 squared to get 1, 24 minus 25 to get negative 1, negative 1 squared to get 1, 18 minus 25 to get negative 7, negative 7 squared to get 49, 29 minus 25 to get 4, 4 squared to get 16. Now we need to calculate the standard deviation, which remember is the square root of the summation of xi minus x bar quantity squared over n minus 1. So we need to sum up this column, add all those up. When we sum them, that sums up to be 76. So our standard deviation is going to be equal to the square root of 76 over the sample size minus 1. 5 minus 1 is 4. Calculate this out. We end up with a standard deviation of 4.36. And finally, we need to get the standard error of the mean, which again, remember, is the standard deviation divided by the square root of the sample size. So for this particular sample, the standard deviation is 4.36 divided by the square root of 5. Calculate that out. We get a standard error of the mean of 1.95. All right, so now let's go back to the question for a second. So this particular question now says, please create a bar graph that illustrates the means of the three groups to within a 95% confidence interval. So let's do that here. So we're going to graph on our y-axis the mean height. Put our units, we're measuring that in centimeters. And then down here, we're going to graph fertilizer concentration. All right, so I'm going to go over, let's say 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Since I don't really have specific units here, I'm just putting some space. I want to graph the means here. So I want to graph, I want to put some spaces between my bars, and that's why I made those dots and spaced them out 4 units apiece. So first of all, I want to have 20 grams per liter, 40 grams per liter, and 60 grams per liter. And actually, let's do this way. Let's think about this way. So let's say that this was 5, 10, 15, 20. So it would be 20, 25, 30, 35, 40 would be right there. 45, 50, 55, 60. So we're making each one of those be grams per liter, 5 grams per liter. So the first thing we want to do is graph the means. So for the 20 gram per liter sample, our mean was 15.8. For the 40, it was 21.2. And for the 60, it was 25. So maybe a good way of graphing this, if we made each one of these lines be 2. So this could be 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30. That should be far enough. So for the 20 gram per liter sample, our mean is 15.8. So we're going to come over here. We're going to draw up a bar to about 15.8. Maybe like so. For the 16 gram per liter or 40 gram per liter sample, 21.2 is what we're looking at. So we need to go up to 21.2. Maybe somewhere right in there. Like so. And then finally for 60, we're graphing a mean of 25. And again, mine's going to be kind of messy since I'm doing this on this computer screen as we go. But you get the general idea. So you would want those to be nice straight bars. So now we've graphed the means. I'm going to go ahead and label this mean as 15.8. This mean as 21.2. And this mean as 25. Now, to do the 95% confidence interval, a 95% confidence interval basically says if you start with a mean and you go up 2 standard errors of the mean from that mean, and you go down 2 standard errors of the mean from the mean, you've created this interval between which you can be 95% sure that the true population mean can be found. So we know for each of these samples, our sample mean. That was based on just a few plants out of huge populations. By doing this standard error of the mean calculation and creating these confidence intervals, we're projecting some upper and lower boundaries for where we can be 95% sure that true population mean will set. So what we need to do first is go to the 20 gram per liter sample. Our standard error was 2.01. So we need to go up and down from that 2 of those, 4.02. So our mean here was 15.8. So we need to go up and down from that 4.02. So if we go up from 15.8, if we go up 4.02, that's going to put us at 19.82. So that would put us somewhere right in here. We're going to make a bar like so. And then we're also going to go down 4.02. So if we go 15.8 minus 4.02, that puts us at 11.78, which should be somewhere right in here. We make a bar there. So what these little error bars are telling us is that for the 20 gram per liter sample, which by the way, we need to label down here, we can be 95% sure that the entire population mean lies somewhere between this bar at the bottom and this bar at the top. So somewhere between 11.78 and close to 20. Let's do the same calculation for the 40 gram per liter sample. The standard error of the mean for that one was 1.85. So let's go 1.85 times 2. That's 3.7. So we need to go to our mean right here and go up 3.7. So that's going to put us at 24.9. So that would put us somewhere in this area. Make a bar. Like so. And we also need to go down 3.7. So 21.2 minus 3.7. That's going to put us down to 17.5, which would put us somewhere right in here. So we can be 95% sure that for the 40 gram per liter sample, our true population mean is somewhere between this bar and this bar. Finally, let's do the same thing for the 60 gram per liter sample. There we had a standard error of the mean of 1.95. So let's take 1.95 times 2. That's 3.9. So we need to go up 3.9 from 2.5, the mean. So that would put us at, from 25 rather, that would put us at 28.9. So that would put us somewhere right in here. Like so. And then we need to go down 3.9 from 25. And that's going to put us at 21.1. So that would put us somewhere right in here. So those are our error bars. Now, the rest of the question, though, wants to know, based on the sample means and the SEMs, identify the two populations or groups that are most likely to have statistically significant differences in height. Justify your answer. The important thing to look at is the overlap of the error bars. If the error bars overlap between two groups, they're likely not to be statistically different. So notice that there's overlap here and here. Notice the overlap between the 60 gram and the 40 gram per liter sample. Also notice the overlap between the 20 and the 40 gram per liter sample. But the two that don't overlap at all in terms of the error bars are the 60 and the 20. And since they don't overlap, we can be somewhat sure that the 20 gram per liter sample is statistically different than the 60 gram per liter sample. We'd probably actually need to do some more sophisticated statistical tests to be sure. But for right now, and for terms of AP biology, if the error bars between two groups do not overlap, you can assume that the two groups are statistically different in a significant way. If the error bars do overlap, you can determine that the differences between the two groups are not statistically significant. All right, so now we have a couple of examples from AP exams in the last couple of years. So here's one about trichomes, the little hair-like structures that grow on fast plants. And we're looking essentially at the density of trichomes within a different group of plants. We have three populations. So what we've done is we've calculated the mean for each of those three samples. We've also calculated the standard error of the mean for each of the samples. So first of all, it says on the axis provided, created an appropriately labeled graph to illustrate the sample means of the three populations. So let's go to a piece of graph paper. What we're graphing again are the population against the number of trichomes per square centimeter. So here we're going to put number of trichomes per centimeter square. And down here, this is going to be population number. Now we have three populations, 1, 2, and 3. So it doesn't really matter where we start with these. I'm just going to space them out about 4 lines apart. So this is going to be population 1, 1, 2, 3, 4. This is going to be population 2, 1, 2, 3, 4. And this is going to be population 3. So we want to graph the means first of all. So we have means of 9, 11, and 14. So let's see. Let's just make these be 1 apiece. So 1, 2, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. That should be enough. So again, we have means of 9, 11, and 14. So for population 1, we're going to go up to 9, and we're going to make a bar graph. For population 2, we're going to go up to 11, and we're going to make a bar. And for population 3, 14. Up to 14. Make our bar. And again, hopefully we won't have curvy lines on our real graph. Next, we want to make a 95% confidence interval. And remember, to do that, we need to go up and down from the means 2 standard errors. Well, our standard error for all 3 of these samples is 1. So if we go up 2 standard errors from 9, that would put us at 11. We'll put a bar there. If we go down 2 standard errors from 9, that puts us at 7. So we can be 95% sure that for population 1, the mean number of trichomes per square centimeter is somewhere between 11 and 7 for the true population. For population 2, again, we need to go up 2 standard errors. So from 11, if we go up 2 standard errors of the mean, that puts us at 13. If we go down, that's going to put us at 9. From population 3, up 2 standard errors is going to put us at 16. Down 2 standard errors is going to put us at 12. So now we have our error bars. So for population 3, we can be 95% sure that the true population mean for number of trichomes per square centimeter is somewhere between 16 and 12. So the next part of the question says, based on the sample means and the standard errors, identify the 2 populations that are most likely to have statistically significant differences in mean stem trichome density. So notice that these 2, population 2 and 3, the error bars overlap. That means that those 2 are probably not statistically different from each other, at least in a significant way. Also notice that 1 and 2, the error bars overlap. They're not statistically different. But notice that there is no overlap between 1 and 3. So those 2 are most likely to be different. So essentially you would say 1 and 3 are most likely to be different because their error bars do not overlap. And that's all it takes to answer that question. Here's one final example involving error bars. So this question is about adult male guppies, and we're looking at the number of spots that they have. It says in a study of selection, male and female guppies from diverse populations were collected from different streams and placed together in an isolated environment containing no predators. The study population was maintained for several generations in the isolated area before being separated into 2 groups. One group was moved to an artificial pond containing a fish predator, while a second group was moved to an artificial pond containing no predators. The 2 groups went through several generations in their new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined as shown in the figure below. Vertical bars in the figure represent 2 standard errors of the mean, or SEMs. So for part A, describe the change in genetic variation in the population between 0 and 6 months and provide reasoning for your description based on the means and the standard error of the mean. So between 0 and 6 months. Notice at time 0, our error bars were huge. That means the standard error of the mean was large. And remember, the standard error of the mean was calculated by taking the standard deviation divided by the square root of the sample size. So if the standard error is large, that means the standard deviation is large. That means at time 0, we had a large variation. But notice as time goes on up to 6 months, notice those error bars at 6 months are much smaller. That means the standard deviation is smaller. Therefore, at 6 months, there is less variation. So, first of all, you're going to say that between 0 and 6 months, the amount of variation decreased. And essentially, you're going to say that because at time 0, the error bars were very large, which implied a large standard error of the mean and a large standard deviation. But at 6 months, the error bars are small. That implies a small standard deviation and a small standard error of the mean. And that's essentially all it takes for part A. Now for part B, this is a little evolution review. Propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 20 months in the absence of a predator. Well, this is the absence of a predator line. And notice when there's no predator present, the number of spots increases. What they want you to talk about on part B is sexual selection. That when there's no predator present, females are selecting males with more spots. And because they have more spots and because they attract more females, they reproduce more, and therefore the number of spots is going to increase in the population. On the other hand, part C says, propose an evolutionary mechanism that explains the change in the number of spots in the presence of a predator. Well, that's this line. And notice that when a predator is present, the number of spots goes down. That's because not only do spots attract females, they also attract predators. And that the fish with more spots get eaten more often. Therefore, they don't reproduce as much. And therefore, the genes for lots of spots become less common. The genes for not as many spots become more common. So what they wanted you to talk about on Part C was natural selection. That spots attract predators. Fish with more spots get killed earlier, and therefore they don't reproduce as much as fish with less spots. All right, let's move on to a different topic. This time let's talk about hypothesis testing. So we all know that a hypothesis is a statement that explains some kind of causal relationship between variables. Often after you do some observations, you might want to make a hypothesis for the phenomenon. We're going to call that your working hypothesis. But you can't ever really absolutely prove a hypothesis. So we do this process called statistical hypothesis testing to try to reject what we call the null hypothesis, or H naught. The null hypothesis is essentially a statement that says there's no underlying factor or variable, that the underlying factor or variable is independent of the observed phenomenon. There's no causal relationship between the variables. Usually, what we mean by that is that there's no difference or no association between two variables. For example, maybe we're testing the idea that sugar makes kids hyperactive. So our null hypothesis might be there's no difference in the amount of time that kids given a sugary drink and kids given a sugar substitute are able to sit still. That's our null hypothesis. Sugar doesn't affect how long the kids are able to sit still. On the other hand, we might have an alternative hypothesis that there is a difference between the amount of time kids given sugar and kids not given sugar can sit still. We're going to do a test to either reject or fail to reject the null hypothesis. If we reject the null hypothesis, that's saying there is a difference. So that's a way to support our alternative hypothesis, or HA. So there's several different types of tests we can do to do hypothesis testing. The test you're going to use most commonly in this class is the chi-square analysis test. This compares the data in an experiment. It compares the data we expected to find with the data we actually got. We're going to say that it compares the observed and expected data. It's a way to evaluate the variability that's present in the real world and get an idea if the differences between our real and our expected results are due to chance or to something else. Chi-square is used when we're comparing two or more categories of data. Usually it's going to be comparing categories that we count or have percentages in. In AP Bio, common places to use chi-square are when we're analyzing the results of a Mendelian cross. Maybe we're comparing gene frequencies from Hardy-Weinberg. Maybe we're counting the number of pill bugs that chose a wet environment over dry. Maybe we're counting colors of M&Ms to see if they match some expected distribution. We also can use a t-test. A t-test is used when we're comparing the means to see if the means of two populations differ significantly from each other. This might be really good in AP Bio when we're comparing the means of a number of trichomes in our artificial selection lab. We won't actually ever have to calculate an ANOVA, which is an analysis of variance. But this is a test that can allow us to compare the means of two or more independent, unrelated groups. All right, there are two different ways to interpret the meaning of these statistical tests. One way is to compare our calculated statistical value to a critical value that we'll get from a statistical table. We'll need to use a chi-square or t-test table, like we said, to find this critical value. To use these tables, we need to know our degrees of freedom. The degrees of freedom are pretty much always equal to our number of phenotypes or categories minus 1. Now that can get a little bit more complicated in the t-test, and we'll talk about the t-test later. But for the chi-square, the degrees of freedom are number of phenotypes or categories minus 1. Now in biology, we pretty much always use the 0.05 significance level. Sometimes this is called the alpha level. And essentially, it's the probability of rejecting the null hypothesis when in reality it's true. The significance level is sort of like an error range, an accepted error range. So when we're doing a chi-square test or a t-test, using the critical value method, we're going to compare the critical value to our calculated chi-square or t-test statistic. If the chi-square or t-test statistic is greater than that critical value from the table, we reject the null hypothesis. This means that the differences between our two groups is significant and it can't be overlooked. If, on the other hand, the chi-square or t-test statistic is less than the critical value, we fail to reject the null hypothesis. And this essentially means that the differences between our two groups are small, and there's a chance that those differences are simply due to chance. The other way to make sense of these statistical tests is by using a p-value or a probability value. Here we're going to use a distribution table to find our calculated chi-square or t-test value. We're going to make sure that we look in the row for the correct number of degrees of freedom. And then we're going to use that cell and evaluate the column above it to estimate a p-value. The probability of whether the results of an investigation differ from the null results by chance alone is the p-value. A p-value of 0.05 means that there's a 5% chance that the difference between the observed and expected data is a random difference, simply due to chance, and a 95% chance that the difference is real and repeatable. That's what we consider significant. Anything below that. So if an investigator's p-value is greater than 0.05, we're going to fail to reject the null hypothesis because there's a chance the differences could be simply due to chance. On the other hand, if the p-value is less than 0.05, we're going to reject the null hypothesis. And this indicates that the differences between the observed and the expected data are significant, and it's not likely that they're due to chance. This same p-value approach can be used with t-test and ANOVA testing. P-values less than 0.05, we're going to reject the null hypothesis. There is a statistical difference. P-values greater than 0.05, we're going to fail to reject the null hypothesis, and this means there's a good chance that the differences between our groups are due to chance. So here's an example. So we have a student who wanted to know if pill bugs preferred wet or dry environments. He set up a choice chamber with a wet and a dry side. He placed 10 pill bugs on each side of the chamber, and after 2 hours, he found 14 on the wet and 6 on the dry side. So his null hypothesis was that there's no preference for wet or dry. They don't care. That's his null hypothesis. His alternative hypothesis is that pill bugs, they like wet better. They prefer wet. So his phenotypes are wet and dry. His expected values on each side are 10. That's what he would expect if there was no difference. But his observed values are 14 and 6. So since we're counting things here, this would be a good time to do a chi-square analysis. So remember, our formula for chi-square looks like this. To calculate the chi-square value, we're going to set up a table. And our table, we're going to do our phenotype, our observed value, our expected value, O minus E, O minus E squared, and finally, O minus E squared all over E. So our phenotypes or categories here were wet and dry. Our observed were 14 in the wet, 6 in the dry. We expected, according to our null hypothesis, 10 on each side, 10 pill bugs on each side. So now we're going to just fill in the table. Next column is O minus E. So 14 minus 10 is going to be 4. 6 minus 10 is going to be minus 4. O minus E squared is going to be 4 squared, 16. Negative 4 squared, 16. And then in the last column, we're going to take 16 divided by the expected of 10, 1.6. 16 divided by the expected of 10, 1.6. Remember that chi squared is equal to the summation of O minus E squared over E. So that tells us that to find chi squared, we simply need to add up those values. So chi squared equals 3.2. So this is our chi-square statistic. Now we need to compare that to a critical value or use it to find a p-value. So let's use the critical value approach first. So here is a chi-square table. In the example that we just worked, we had either wet or dry, 2 categories, which means we have 1 degree of freedom because our degrees of freedom are 1 less than our number of categories. We work at a probability level of 0.05. So our critical value was 3.84. That came from our table. Again, our chi-square statistic was 3.2. All right, so in this case, our chi-square value is less than our critical value. In that case, we're going to fail to reject the null hypothesis. And what that means is that we expected 10 and 10. We ended up with 14 and 6. Yeah, there's some differences, but those differences are relatively small, maybe due to chance. The differences aren't big enough to be statistically significant. All right, so let's use the p-value approach. So again, our chi-square value was 3.2. We had 1 degree of freedom. So we're going to go in this chi-square distribution table, different table this time. We're going to go to 1 degree of freedom, and we're going to find 3.2. Notice that 3.2 would be somewhere in here. And if we go up from that 3.2 right in here, and we look up to the p-values, which are the p-values up here, notice that our p-value is somewhere between 0.1 and 0.05. Point is, our p-value is greater than 0.05. When p-values are greater than 0.05, you're going to fail to reject, some people say accept, the null hypothesis. We're saying that the differences between our observed and expected data are small and not big enough to be statistically significant. All right, here's an example from an AP exam. So in this case, we have some plants that are sprayed with an insecticide. We also have a control where the plants, some of the other plants are sprayed with only water. Now it's important that we don't just have dry plants because we want to factor in the effect of the water. So we have one set of plants sprayed with water, one set of plants sprayed with insecticide. And essentially what we want to know is during what part of the life cycle, if any, does the insecticide spray make a difference? So we're looking at essentially the mean number of plants in a certain area, and we compare them. So for example, during the flowering plant stage, there were this many plants in the water spray, this many in the insecticide spray. Yeah, there were more in the insecticide sprayed area than there were in the water sprayed area, but not that many more. And the p-value was greater than 0.05, which means these differences are not statistically significant. In the next group, the seed formation stage, 2440 here, 3100 here. Different, but not that big a difference. Again, the p-value was greater than 0.05, so the differences were not statistically significant. But in the next 2 stages, notice the differences between the insecticide sprayed and the water sprayed are pretty significant. and the p-values are less than 0.05. So what that means is that the differences here are statistically significant, whereas the differences up here, not so much. All right, that's a quick overview of hypothesis testing, descriptive statistics, and inferential statistics. Hope that helps.